Hello students, welcome back to your own channel Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. So before we start today's amazing class, quickly let me know in the chat if my voice is audible to all of you. And the presentation behind me, the beautiful DNA, which is a nucleic acid, which is a biomolecule, visible to all of you. Quickly let me know. Yes Niharika, yes Jai, yes Neha, Madhumita, Naina, Sangeeta, Meghana, Navindrani. Yes, good morning to each and every one of you. Now, let's start the class with a basic recap of what we did in yesterday's class. Now, this is a part 2, all of you know. Movie has part 1 and part 2. This is a simple movie which has a part 1 and part 2. In yesterday's class, we started with some basic experiments. Then we did about amino acids. Then we did about nucleic acids. Then we also did about lipids and secondary metabolites. All of these topics in yesterday's class. Now, what will we do in today's class? What is the importance of today's class, students? Because the most difficult part of this chapter, which everyone faced difficulty in, the enzyme part, the substrate concentration, what is this Vmax, what is half Vmax, what is Km value, all of those topics in today's class. But we'll be starting with some basic questions. We'll be starting with a basic concept, that is carbohydrates. Okay, so we'll be starting the class with carbohydrates, then we'll be going to other complex chapters, other complex topics. So quickly let me know in the chat if you're ready for today's class, because it's going to be a blast. It's going to be amazing. I have a lot of questions. I have NCERT snips for you, concept, everything at one place. Clear? So can we start, students? Yes, we can start, definitely. I can see some green hearts in the chat, which is obviously the most amazing part, right? The green hearts in the chat, which obviously gives us motivation. Okay, so let's start with a basic concept. Let's with a basic question first of all. Actually, here's a question, basic question for all of you. Let's see how many of you are able to solve this question. It says that which of the following statement is incorrect about an enzyme? There are 4 options here. Don't worry if you do not know. We'll come back to this question once we are in the enzyme part. This is just what do I say? To activate your brain cells, I have added a question here. Don't worry, by the end of today's class, you will be able to solve such NEET 2024 questions with easily. Okay, so we'll come back to this question later on. Remember this question before we start the enzyme concept. Okay, now students, the first topic for today is nothing but carbohydrates. It is going to be carbohydrates. As the name suggests, carbohydrates. What are they? They are hydrates of carbon. They are hydrates. They are hydrates of carbon. Hydrates of carbon. Now, if you look at the basic structure, of any type of carbohydrate, do you notice multiple OH groups everywhere? Multiple OH groups? Yes. So they will have multiple OH groups. Okay. Now, apart from that, in an aldehyde, you can usually find something called as aldehyde and a ketone. In the case of your carbohydrates, you will find an aldehyde or ketones. Okay. You will find aldehyde or you can find ketones. Clear? Now, aldehyde you can find in the form of sugar. Can you see this is the aldehyde group? In the case of fructose, we can find the ketone group. Ketone group in the case of fructose, and aldehyde group in the case of glucose. And glucose and fructose, which you have been studying throughout your 11th and 12th journey, your glucose and fructose are part of carbohydrates. They are part of carbohydrates. Clear? Now, very simple. The next thing we need to understand the classification of carbohydrates. Now, the classification of carbohydrates is based on the number of sugar. Present. For example, in the case of monosaccharide, mono means one, saccharide means sugar. So if there is a single sugar, it is called as monosaccharide, right? Monosaccharide. Now students, what about disaccharide? Di means what? Di means 2. Saccharide means sugar. So there are 2 sugars joined together by a bond, glycosidic bond, that is disaccharide. Then we have similarly oligosaccharides as well as polysaccharides. Poly means many. Poly means many. When many sugars are joined together by a glycosidic bond, that is called as polysaccharide. That is called as polysaccharide. Clear? Now students, now carbohydrates we learned monosaccharides, disaccharides, oligosaccharides. What is monosaccharide? It is the simplest sugar, forms the building blocks of large carbohydrates, that is many monosaccharides join together and they make a polysaccharide. Clear? So we have monosaccharide 1, monosaccharide number 2, monosaccharide number 3. When many monosaccharides join together, they make up the polysaccharide. They make up the polysaccharides. So all of you now know how are the polysaccharides made. All of you have an idea about how the polysaccharides are made. When many monosaccharides join together like a train, they make up the polysaccharide. They make up the polysaccharide. Examples given here, simple examples: triose, tetrose, pentose and hexose. All you can remember is triose. Easy way to remember, triose is a 3-carbon atom. Remember that. Now what about disaccharide? I told you disaccharide is made up of 2 different sugars. 2 different sugars are joined together. 2 sugars are joined together. Now simplest example is going to be sucrose, which all of you know. Sucrose is made up of glucose, 1 sugar, fructose is one more sugar. When 2 sugars are joined together with the help of glycosidic bond. Which bond? Glycosidic bond. Glycosidic bond, it is called as disaccharide. It is called as disaccharide. 2 sugars are joined together. One more example is lactose, which is present in the milk, which is made up of glucose is 1 sugar, galactose is the 2nd sugar. 2 sugars are joined together with the help of glycosidic bond. 2 sugars, disaccharide. Disaccharide. Clear? It's called as disaccharide. All of you in the chat, quickly tell me. All of you should type down in the chat. Sucrose, glucose plus fructose, lactose, glucose plus galactose. Easy. Easy. Right? No. Next one is oligosaccharide. Oligosaccharides are basically more than 3 to 9. When we have 3 to 9 monomeric units, when there are 3 to 9 monomeric units joined together, that is called as oligosaccharide. The only example you have to remember is raffinose. Raffinose is the only example. It is made up of 3 different monosaccharides: glucose, galactose and fructose. 3 different sugars joined together, raffinose. Done. So next one is polysaccharides. Okay, this is why does it look like one? Okay, friends, there is one something weird thing is happening here, but I will draw it in a better manner. Okay, I think there is one alignment issue here, but don't worry, I'll tell you what's there in that. Now polysaccharides, obviously, what is the meaning? Poly means many. When many monomeric units are joined together, they make up the polysaccharides. But polysaccharides are actually of 2 types. Polysaccharides are actually of 2 types. For example, all of you are in class, imagine some of you are in class 11th, some of you are in class 12th. Okay, some of you are in class 13. Okay. Now, imagine I ask all the class 10 students to stand in one line. I ask all class 11 students to stand in one more line, and class 13 or class 12 students to stand in one more line. Now everyone is standing based on their class, which class they are in. Is there any mixing up? No. Every student in one line is class 10th. That is called as homogeneous, but everyone is in class 10th. Similarly, we have something called as homopolysaccharides. We have something called as homopolysaccharides. In the case of homopolysaccharides, in class 10th, how we had all the students from the same class. In the case of homopolysaccharides, all the sugar units are same. All the sugar units are same. For example, in the case of cellulose, we have long chain of glucose. Glucose is standing. Long chain of glucose. Everyone is glucose there. Everyone is glucose. See, in the case of chitin, long chain of NAG. N-acetyl glucosamine, NAG molecules, NAG. Many repeating units of NAG. See, in the case of starch, Again, there is amylose and amylopectin. Yes, amylose and amylopectin is there. Students, this is looking little cursed. One second, students. I will add a separate slide. One second. We go back. One second, let's go back. So, polysaccharides is here. Polysaccharides broadly classified into two categories. We have homopolysaccharides and heteropolysaccharides. Heteropolysaccharides. Okay. Homopolysaccharides are again divided into two categories based on structure here. See, based on structure and based on storage. Structure and storage. Based on structure and based on storage. In structure, we learned about cellulose and we learned about chitin. Cellulose in the case of plants, chitin in the case of insects. The outer shell of insects or arthropods. You can tell anything, same thing. Insects. Outer shell of insects. And also fungus also. Now under storage we have starch, glycogen, inulin. Starch, glycogen as well as inulin. I am writing insulin. Inulin. Now in heteropolysaccharides, two things you should remember, mainly two things. The first one is peptidoglycan. Which is found in the case of bacterial cell wall, peptidoglycan, as well as agar-agar. Just remember this, you are set to go. Clear? Now here, cellulose and chitin, they are present in the structure of a organism. Storage, starch in the case of plants, glycogen in the case of animals, inulin also in the case of plants. Okay. Now inulin, all of you should know, I told you, inulin is a type of homopolysaccharide. It is homopolysaccharide. So it should have repeating units of only one type of sugar, right? Only one type of sugar should be there. In the case of inulin, it is not glucose. In the case of inulin, it is fructose. It is going to be fructose. Long chain of fructose. Fructose. One line will be there, fructose. Like how we have class 11 standard line, height wise. Similarly, we have fructose joined together. Homopolysaccharide. Now, what if we mix? Now? On Saturdays, do you stand according to the house wise? Like blue house on one side, red house on one side, yellow house on one side. When you are standing house wise, do you mix class 10th, 11th, 12th? Yes, you mix. You mix that time, right? All the red color house will be in one. But the red house will have class 10 students, class 11 students and class 12 students also. That is your heteropolysaccharide. In the case of heteropolysaccharides, we have mixing of sugars. For example, in peptidoglycan. Peptidoglycan is made up of NAG and NAM. Yes. N-acetyl glucosamine, N-acetyl muramic acid. So NAG, NAM. That is heteropolysaccharides. Many polysaccharides, but they are different sugars. Agar it's not in your syllabus, but I'll mention later on. Okay. See here, property students, this is a very important slide for all of you. For example, starch, polymer of glucose, we learned that. Food reserve in the case of plants consists of amylopectin and amylose. This you will learn in chemistry biomolecules. This you will learn in chemistry biomolecules. There is something called as starch is made up of amylopectin and amylose. Amylose is alpha 1,4 linkage and amylopectin is alpha 1,6 linkage. That is branching. Long chain branching is there. Okay. Then glycogen. Glycogen is a polymer of glucose, again same. Food reservoir in the case of animals. That is the main difference. The other structural difference between starch and glucose is going to be, in the case of glycogen, the alpha 1,6 linkage is much more higher. It's more branched. It is highly branched. Highly branched. Okay. Highly branched. Sangeeta, all the best. It is stored in liver and muscles. Now, inulin, I told you, inulin in the case of plants, it is a polymer of fructose. NEET PYQ. Here is a NEET PYQ, students. NEET PYQ. Inulin is made up of long chain of fructose. Long chain of fructose. Okay. Not metabolized in human body, stored in dahlia and dandelions and artichoke. It is present in the case of plants. Now, what about cellulose? All of you know cellulose. All of you know cellulose. It is a polymer of glucose. Forms the structural part of the cell. That is cell wall. Cellulose is part of cell wall. Also found in the case of algae and everything. Okay. Straight chain, unbranched. Not unbranched. Not like, it's like very alpha 1,6 linkage is more. Chitin is a polymer of N-acetyl glucosamine. It is NAG. Long repeating units of NAG. See, all of these are homopolysaccharides. Okay. Exoskeleton of arthropods. That is insect. Insect will have an outer covering on top. Yes. The outer covering is basically made up of chitin and it is present type of homopolysaccharide. They'll ask you a question, direct questions. Chitin is a homo or heteropolysaccharide? You should be able to tell, sir, chitin is a homopolysaccharide. Okay. It's a complex polysaccharide, but it is a complex polysaccharide. Now, what about heteropolysaccharides? I told you peptidoglycan. Peptidoglycan is made up of NAG and NAM. It is made up of NAG and NAM. Two repeating units. NAG and NAM. Remember, that is peptidoglycan. For agar, you don't have to remember this. It is not given in NCERT, but I have given you for your, say, like just better understanding. It is made up of galactose and 1,3 anhydro-L-galactopyranose. Obviously, they will never ask you all this in class 12th level. They will obviously ask you this in MBBS level. MBBS level they will ask you, but not right now. Okay. And remember, students, agar is obtained from your red algae. Red algae is also called as chlorophyceae or pheophyceae or rhodophyceae. It is also called as rhodophyceae. So it is found in the case of rhodophyceae. Okay. Gelidium and Gracilaria. Any doubt here, students? I believe it's very easy, right? There is no very difficulty level here. It's very easy. And I hope you're able to understand this, right? Homopolysaccharide, heteropolysaccharide. Examples are extremely important. Question will be directly asked from the examples and there is no shortcut to this. There's no shortcut to this. Okay? No shortcut to this. Now, you might have heard about this concept somewhere. Reducing sugar. You might have heard about sucrose is not a reducing sugar. Even though sucrose is a disaccharide, it is not a reducing sugar. You might have encountered this question so many times in so many test paper, mock paper, everywhere. But what is reducing sugar, students? What is reducing sugar and what is the importance of reducing sugar for us right now? What is this reducing sugar? Right? You might have heard about it. You might have not heard about it. Right? What is this reducing sugar? It's a very easy concept. You might have heard about this from the chemistry labs, right? You might have done this experiment in your chemistry labs, that Benedict solution, and you take it in a test tube, add the Benedict solution and completely, keep it in a hot water bath. There will be a color change. Let's understand what is reducing sugar right now. Okay. So first important point about reducing sugar is that all the reducing sugars, mostly your monosaccharides and few disaccharides, all of these reducing sugars have a free aldehyde. They have a free aldehyde or a ketone group. Free aldehyde or a ketone group where reactions can take place. For example, here, look at here. Glucose, free aldehyde group. Fructose, free ketone group. Galactose, a free aldehyde group. All of these are monosaccharides. All of these are monosaccharides. They have a free aldehyde or a ketone group. Point number one, clear. This is the point number one. Point number one. The next thing you need to understand, here is that reducing sugar, you take 2 ml of test solution, which is glucose. 2 ml of glucose is here. 2 ml of glucose. Okay, taken, sir. 2 ml of glucose taken. In this 2 ml of glucose, you need to add equal amount. If you're taking 2 ml here, 2 ml of Benedict solution. So when you take 2 ml of Benedict solution, what exactly happens? There is a reaction. There is a reaction, that is reducing sugars reduces the cupric ions. Reducing sugars such as your glucose, fructose and galactose. The reducing sugar will reduce the free cupric ions to cuprous ions. Cupric ion to cuprous ions in the Benedict solution. All monosaccharides reducing sugar. And when you heat it in water bath, the Benedict solution will turn into brick red. Brick red precipitate can be obtained. That is your reducing sugar. Reducing sugar has an ability to reduce free cupric ions to cuprous ions. Now more about this reactions you will learn in chemistry. This is where I will teach stop here. Okay, let's not go deep into it. Now, I told you here monosaccharides are reducing sugar. Even students, even disaccharides are reducing sugar, but not all of them. For example, sucrose. Sucrose may be a disaccharide, but sucrose is not a reducing sugar. Similarly, polysaccharides. Poly and oligosaccharides are also not reducing sugars. You might ask me, sir, why are they not reducing sugars? They are not reducing sugars because there is no free aldehyde or ketone there. There is no free aldehyde or a ketone there. Done. Students, do you want to do one? See this, non-reducing sugars such as your polysaccharides. Even the sucrose is the disaccharide. It does not have a free aldehyde or ketone. No free aldehyde or a ketone here. So non-reducing sugars. Non-reducing sugars and no free aldehyde or ketone. And students, remember, a polysaccharide will have a reducing end and non-reducing end. That is why they are non-reducing sugars. Okay. Now students, do you want to do one more experiment? Do you want to conduct one more experiment right now? Tell me yes or no in the chat. One more experiment now. One more experiment you want to do? One more experiment is very simple. Second experiment is again very simple. Again, you might have done this experiment in your classes, in your chemistry lab or biology lab or anywhere. You might have done this experiment. In the second experiment, what you need to do is, you need to take iodine solution. You have to add the iodine solution to a starch containing sample. Done. At the same time, you need to take iodine solution, add the iodine solution into a cellulose containing sample. Here we have starch, here we have cellulose. Here we have starch and here we have cellulose. Iodine is actually somewhat brownish or yellowish color. If it's very diluted, it's very yellowish. If it's very concentrated, it's going to be brownish color. So when you add iodine to starch and when you add iodine to cellulose, both of them react very differently. The main principle behind the reaction lies in the structure of cellulose and starch. See, for example, if you look at the structure of starch, it has a lot of branching here. Can you see? Amylopectin, for example, lot of branching. At the same time, if you look at the structure of cellulose, it does not have any branching. Beta linkage, alpha 1 to 4 beta linkage, cellulose structure. Now tell me, if for example, I am able to hold this pen with my hand. Why? Because I have fingers. Because I have fingers, I'm able to hold this pen with ease. So this branching is there, right? This branching can actually hold the iodine molecules. They can hold iodine. What about cellulose? Cellulose does not have any branching. So can it hold iodine? Obviously not. If there are no fingers, if I do not have any fingers, can I hold the pen? No, obviously not. I cannot hold the pen. Right? So similarly, how cellulose does not have any types of branching, it cannot hold I2, it cannot hold iodine, and that is why, see here students, when you add iodine solution to cellulose, it will remain brown color. But when you add iodine solution to starch, it will become blue color. It will become blue color. Clear? That's why. Why? Because starch can hold I2 molecules in the helical portion. This right here is the helical portion. This right here is the helical portions here, branching. They can hold I2 here. I2 can be holded here. Iodine can be holded here. So positive test is blue color. But cellulose does not contain complex helicals because it is linear. It is completely linear in shape. There is no complex helicals here. Complex helicals are completely absent in the case of cellulose, right? And they cannot hold iodine molecules, so negative test, that is brown or yellow color. Brown or yellow color. Simple. Simple. Done? Understood? Yes. Beautiful. Now see this question. This right here is a NEET PYQ. It's a NEET previous year question for all of you. It's a NEET PYQ question. Look at the question. It says that cellulose does not hold blue color with iodine because it breaks down when iodine reacts with it. It is a disaccharide. It is a helical molecule. Cellulose does not contain complex helicals and hence cannot hold I2 molecule. That's right. Cellulose does not have any type of helical and it cannot hold I2. It cannot hold I2. So it will become brown color. It will remain brown color in the case of cellulose. Cellulose. Understood? Understood all of you? Yes. Now look at this question. Look at this question. I think I was going to give this as homework question, but let's solve it. Now protein, what do we learn? In the case of protein, we have the peptide bond. Yes, peptide bond. Unsaturated fatty acids will have double bond because saturated means single bond. Unsaturated will have double or triple bond. Right? Nucleic acid will have phosphodiester bond. Yes. And polysaccharides, we just now learned, polysaccharides have glycosidic bond. Polysaccharides have glycosidic bond. We just learned that in today's class. Right? Polysaccharides, glycosidic, proteins will have peptide bond, nucleic acid. In yesterday's class we learned. In yesterday's class we learned how nucleotides are attached. Nucleotides are attached, that is 3 to 5 phosphodiester linkage. So what is the answer? A is 4, these two are gone then. These two are gone. A is 4 and B is going to be 1. B is going to be 1, so 3 is the answer. 3 is the answer then. Yes. Look at this question now. Look at this question. Students, all of these questions are NEET PYQ questions. I am not making easy questions for you. I am taking previous year questions and making sure that yes, whatever you learn in the class is actually beneficial to solve PYQs, to solve questions, will help you to understand and master the chapter. But look at this question now. Inulin is a polymer of, we learned it, it is a polymer of fructose. Many fructose are joined together. Many fructose are joined together. Fructose. See this, exoskeleton of arthropods is made up of. Exoskeleton of arthropod is made up of, we just learned now that it is made up of chitin, not cutin, chitin. It is going to be chitin. Done. Right? Next question. Which of the following is not a complex polysaccharide? See, chitin is complex. Chitin is complex, made up of NAG, right? Glucosamine also complex. I told you, didn't I write around chitin? Didn't I write around chitin when we went to chitin? Didn't I mention it is a complex polysaccharide? Here. It is a complex polysaccharide. Chitin is a complex polysaccharide. The points are very clear to all of you to understand the concepts. Yes. And N-acetyl galactosamine also is complex. Glucosamine is also complex. Chitin also is complex. So which is not a complex, that will only and only be inulin. Inulin is just made up of a simple fructose, a simple monosaccharide which is a fructose. Fructose, 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 fructose. Done. And students, many students went wrong here. Can you see? 57% of students went wrong here. 50% of students went wrong here. It's a NEET 2024 PYQ. Done. Now students, let's read the NCERT lines to make sure we have covered every single point. Yes? Can you do that? And I saw yesterday's comments. All of you love it when we read the NCERT and make sure whatever we're studying is there. Right? Proteins are polypeptide. Poly means many, peptide bonds. Yes? We have done this in yesterday's class itself, right? Each protein is polymer of amino acids, such as 20 types of amino acids are there: alanine, cysteine, tryptophan, lysine. A protein heteropolymer and not a homopolymer, obviously, because it is made up of many different types of amino acids. It cannot be homopolymer at all, right? A homopolymer has only one type of monomer repeating n number of times. This information is about amino acid content. Important later you will study in attribute lessons. Okay, what is important here? Amino acids can be essential and non-essential. That is, non-essential, our body makes it. Essential amino acids we get through our diet, right? We learned this yesterday's class. Some transport nutrients, function of proteins. See, function of proteins. Some transport nutrients across cell membrane. Some fight infection, that is antibodies. Some are hormones, and some are enzymes. This is the most important table. Let me write down here. Very important. Very important table. For example, collagen, intercellular ground substance. That is, collagen makes up the complete network. Trypsin is the enzyme found in the small intestine, secreted by the pancreas. Insulin is a hormone, diabetes. Antibodies fight against infection. Receptors, sensory receptors like smell, taste, hormones, everything receptors are made up of proteins. Even GLUT4. GLUT4 is a type of protein which helps in transportation, of glucose into the cell. If glucose cannot enter the cell, glucose will sit on GLUT4 and will enter into the cell. Remove this table right here. Remove this table. Now, apart from this, there is one important lines here, that is, collagen is the most abundant protein in the animal kingdom, in the animal world. Collagen, animal world. Let me write here. Collagen is abundant protein in animal world. That is collagen. Now what about rubisco? Rubisco is the most abundant protein in whole biosphere. In the entire biosphere, it is the most abundant protein. Now, if I ask you a question, which is the most abundant carbohydrate? Most abundant carbohydrate. Most abundant carbohydrate. Most abundant carbohydrate is going to be cellulose. It's going to be cellulose, because cellulose is present in every single plant. It is present in every single plant. So plants covers such a large portion of your land, and we find so many plants on earth. Algae also. Yes, algae also. So it's the most abundant carbohydrate. Remember this, you will get a lot of questions from here, and you will get a lot of questions from here also. Done? See this question right now. See this question right here. GLUT4 is what? This is a NEET 2024 question, by the way. This year question, not anything old or easy question. GLUT4, we just now learnt, enables of glucose into the cell. Yes. Insulin is a hormone. Trypsin is an enzyme. And collagen is a intercellular ground substance. So A is going to be 4. It means option number 4 is the answer. Done. Done. Right? Very easy. Very easy. Now students, let's read the NCERT lines for polysaccharides. Yes, The acid, first of all, polysaccharides will be found in the acid insoluble fraction. Now tell me, acid insoluble is part of filtrate or retentate? In the chat, quickly go. Acid is part of retentate, acid insoluble, retentate or filtrate or retentate. Polysaccharides, their molecular weight is more than 1000 daltons. More than 1000 daltons. Okay. Carbohydrates, another class of macromolecules. Macromolecules. Since it is a macromolecules, it will be in acid insoluble fraction on the top, that is called as retentate on top. Okay. Polysaccharides are long chains of sugars, thread or cotton thread containing different types of monosaccharides. So many monosaccharides come together and they make up the polysaccharide. They make up the polysaccharide. Cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. That is, cellulose is made up of repeating units of glucose, beta 1,4 linkage. Cellulose is a homopolymer. Starch is a variant of this but present in the storage. I told you, cellulose and chitin for structure. Cellulose and chitin for structure. For storage we had cellulose, glycogen and inulin. All three are for storage. Storage homopolysaccharides. See, present as storage, that is starch. Present in the case of plants. In the case of animals we have glycogen. Inulin is a polymer of fructose, which is also found in the case of plants. What is difficult in this, students? Everything is given here. Now, in polysaccharide chain, say glycogen, the right end is called as reducing end. I showed you this actually in one of the diagrams. The right end is always called as reducing end and the left end is called as non-reducing end. Left is non-reducing, right is reducing. Right is reducing end. And left part is going to be non-reducing end. Non-reducing end. Okay. Now, starch forms helical secondary structures. In fact, starch can hold I2 molecule in helical portion, and that is why starch and I2 is a blue in color, and cellulose does not contain complex helicals. Cellulose structure is very linear, cellulose. That's why it cannot hold I2. Experiment which we did. Right? Now, plant cell wall is made up of cellulose, paper is made from plant pulp, and cotton fiber is cellulosic. There are complex polysaccharides also, such as N-lactoglycosamine, glycosamine, exoskeleton of arthropods, for example, have a complex polysaccharide called as chitin, right? Exoskeleton has chitin. These complex polysaccharides are mostly homopolymers. See, they are homopolymers. Chitin is a homopolymer. And N-lactose is glucosamine. All of them are complex polysaccharides. Inulin was simple. That question came from here, this line of NCERT. Okay. Nucleic acids also we did in yesterday's class. Nucleotide we did. They are part of true macromolecules because their weight is much more higher. Yes. We learned in yesterday's class, right? All the different types of different types of nitrogenous bases. Adenine and guanine are going to be purines, as well as your rest, that is cytosine, thymidine and uracil are going to be pyrimidines, right? Deoxyribonucleic acid and RNA. Our DNA will have pentose sugar, it's called as deoxyribose sugar, and ribose sugar in the case of RNA. We learned a structure also in yesterday's class. Look at this question now. Question is very simple. Which of the following is the most abundant protein in the animal kingdom? We just now learned it is collagen. And with that, first part of today's class is over. Now we'll go to the second part, it is protein now. Carbohydrates done. Now I believe there should be no doubt regarding carbohydrates. If there is any doubt, quickly ask me in the chat. Yes. If there is any doubt, quickly ask me. Carbohydrates. Carbohydrate concept done, carbohydrate NCERT reading is done, and carbohydrate questions are also solved. Yes, everything is going like in a smooth butter train. Right, now we'll go to the second part of the class, that is going to be structure of proteins now. Structure of proteins, and again, very easy, you do not have to break your head over this. All you need to remember here is the different structures and how they play an important role. Okay, now the structure of protein can be divided into 4 categories. Entire structure of protein can be divided into 4 different levels. The first level is called as primary structure. Then we have secondary structure, tertiary structure, and quaternary structure. The entire structure of protein is divided into 4 different levels. 4 different levels. For example, this right here is the primary structure of protein. Students, let me tell you, entire notes will be available. These are beautiful notes which will help you to study everything. The notes will be available on the Telegram channel. On the Telegram channel, all the notes will be available to you. So just understand the concept right now. Do not worry a lot. Understand the concept. Immerse yourself. Notes you can get later on if you want. Okay. Now, this is the primary structure of protein. Now, it tells you a significance. There's a significance. What is the significance of primary structure of protein? The main significance of primary structure of protein, it tells you that which amino acid is at which place. That is, position of amino acid is obtained from primary structure of protein. Position of amino acid. For example, we have amino acid 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So primary structure of protein tells you the positions of different types of amino acids, and all the amino acids are linked together by peptide bond here. Peptide bond. They are linked together. Now then we have secondary structure of protein. In secondary structure of protein, there are 2 types. Here it was completely linear. Here. But here the shape changes. For example, this one looks like a solenoid. In physics you might have learned, right? The solenoid structure. Yes. It's also called as alpha helix. The other one is called as beta plated sheets. Let me show you how it looks. Now, you might have played this game when you were kids, right? You need to take a sheet and completely fold it. Imagine you try folding a sheet. Imagine when you fold a sheet like this. Do you get a sheet like this? Have you ever done this? Have you ever done this, students? Something like this? In your life you have done this, right? Folding the sheet like this. This is called as beta plated sheets. They are called as beta plated sheet. Other one is called as alpha helix. Alpha helix will be in a coiled structure. It will be in a coiled structure. Right hand coiling will be there. And here it's important to note that apart from peptide bond, there is also hydrogen bonds here. Hydrogen bonds can be seen here. Hydrogen bonds here. Hydrogen bonds can also be seen here. Hydrogen bonds. Now students, now if you take alpha helix and you take beta plated, if you take alpha helix and beta plated and you make it into a woolen ball like structure, take alpha, take beta, completely mix it and make a woolen ball like structure. That woolen ball like structure is called as tertiary structure of protein. Tertiary structure of protein. And in tertiary structure of protein, it is actually 3-dimensional, 3D. Here it was 2D, everything was 2D. This is going to be 3D, and most of the biological enzymes are coming in tertiary structure. Enzymes are coming here. Okay. And also you can see, apart from hydrogen bond, peptide bond, you can also see disulfide bonds here. In the case of tertiary structure of protein, we can also see disulfide bonds. Now, what is quaternary then? What is quaternary? Quaternary is very simple. You need to take one woolen ball, take one more woolen ball and mix the 2 woolen balls. That is, when we have multiple tertiary structures, take one tertiary structure, take one more tertiary structure, completely mix the 2 tertiary structures. Quaternary we get. Then we end up getting quaternary. Then we end up getting quaternary structures. Can you see? There is 2 chains here. One chain is here, one more chain is here. Chain A and chain B. This is, imagine this is chain A, this is chain A. Similarly, there will be chain B somewhere here. You need to take chain A and chain B and completely mix it together That is quaternary structure. That is quaternary structure. That's all. See here, we learned this. Didn't we learn this? Linear chain of amino acids in the primary represents the position of amino acid. Here there is something called as N-terminal, so C-terminal and N-terminal. C-terminal will have a carboxylic acid. Can you see? There is a C-terminal will have a carboxylic acid on one side. C-terminal. Other one is called as N-terminal. N-terminal will have a amino group. Amino group. That's why it's called as N-terminal. NH2. Okay. C-terminal and N-terminal. Linear. Then we have secondary structures, which is alpha helix and beta plated. This is alpha helix structure, and there will be hydrogen bonding here. Hydrogen bonding here. This is beta plated sheets. Beta plated sheets. Then we have, when we take alpha helix and beta plated, make it a ball-like structure, that is tertiary. 3-dimensional, folding upon itself, biologically active. Biologically active. Done. All the points have been covered. All the points have been covered. Then disulfide bond can be seen there. Quaternary structure. What do they see? Can you see? Peptide 1, peptide 2. Multiple tertiary structures are coming together and they are making the quaternary structures. And here each one is called a subunit. This is subunit, this is subunit. Two subunits coming together, quaternary structure. Two or more polypeptide chains. Now in your NCERT there is an example given. That's right. There is an example given for tertiary structure, that is hemoglobin. Hemoglobin has 2 alpha chains and 2 beta chains. 2 alpha chains and 2 beta chains arranged together, quaternary structure. See here. 2 alpha chains, 2 beta chains. Heme group in the middle. That is the structure of hemoglobin. Hemoglobin is an example of tertiary structure of protein. Tertiary structure of protein. Done. It's very easy to understand. When there is a visualization. There are 4 different chains here. 1, 2, 3 and 4. 4 chains are coming together. They're making up the quaternary structure of a protein. See here, NCERT tells you what? What NCERT tell you? Proteins, as mentioned earlier, are heteropolysaccharides. Heteromers. Heteropolymers. Okay? Primary structure is what? Basically, sequence of amino acid, the positional information of protein is given. That is, which amino acid is in which position. That is telling you basically the primary structure here. Primary structure here. Primary structure here. Okay. Then we have N-terminal will have a amino group. C-terminal will have a carboxylic group. Done. Carboxylic group. Done. The thread is in the form. Then we have secondary structure of protein. See here, only right hand helical are observed, and there is staircase like. Where is the staircase? Only some portions of the protein thread are arranged in a form of helic. That is alpha helix structure. Alpha helix structure. Then the other region of protein thread is now folded into other forms, that is we call the secondary structure of protein. Secondary structure of protein basically has alpha helix and beta plated. Beta plated. Okay. Now long protein chains also fold upon itself and woolen ball-like structure, that is tertiary structure of protein. Okay, which is 3-dimensional, and they are also biologically active. Also biologically active for many proteins. Done. Then we have quaternary structure here. Quaternary structure, that is they make a lot of subunits. I told you many chains coming together, that is called as quaternary structure of protein. Example is going to be hemoglobin, which has 4 chains, 2 alpha chains and 2 beta chains. 2 subunits of alpha chains, 2 subunits of beta chains of the human hemoglobin. That is a quaternary structure of protein. Very easy, right? Structures of protein. Done. Last 1 hour, you have almost done so many topics, students. Because the next chapter, next part we're going to start is nature of bond. We have already discussed this. Peptide bond between the protein. Glycosidic bond is the bond between the different types of polysaccharides. carbohydrates, right? Now look at this question now. Look at this question. Look at this question. The question says that a protein is imagined as a straight line. The left part represents the first amino acid called as C-terminal, and the right end represents the last amino acid called as N-terminal. Adult hemoglobin consists of 4 subunits, 2 alpha and 2 beta. Which is the answer now? Tell me. Which statement is wrong? Which statement is correct here? Which statement is correct? Which statement is wrong? I will show you the confusion, which if you don't read the lines of NCERT, this is where you get confused. See here, if you go back to the NCERT line here, it says here, the left side of represented by the first amino acid. Left side is the first, it is starting from left. Left it is starting. So first is left. The first amino acid, and the right represents the last amino acid. Right is last. Okay. The first amino acid is also called as N-terminal. Remember the diagram. In the diagram, students, in the diagram which we discussed before, left is N-terminal. Starting amino acid is starting with N. Starting with N. Okay? Starting with N. Now if you go to the question, it says that, the question says that the protein is imagined as a line, the left end represents the first amino acid. But is it C-terminal? First left side is N-terminal, right? Isn't it N-terminal? Isn't it N-terminal then? Yes, it is N-terminal. And the right is represented by last amino acid. Yes, right is last. But isn't this C-terminal? Isn't it supposed to be C-terminal? So can I tell statement number 1 is wrong completely? Statement number 1 is wrong completely? Yes, statement number 1 is wrong. Adult hemoglobin consists of 4 chains, 2 alpha and 2 beta subunits. Yes, 2 alpha and 2 beta subunits. That is, hemoglobin consists of 4 subunits, 2 alpha subunits and 2 beta subunits. So this statement is absolutely correct. So what is the answer? 1 is false and 2 is correct. Understanding? Yes. That's how you understand every line of NCERT and frame the questions Okay. And students, if you're joining the session late, quickly smash that like button. Students, I want everyone to quickly smash that like button and like the session, and also share the session with everyone. It's important to share because NCERT line by line, question formation, everything will not happen at anywhere else. Like we have no one clubs everything. Everything is always separate. But we are making sure NCERT reading, concept clearing, PYQs in one session. Right? Look at this question. I think we did this, right? Yeah, we did this. Now look at this question now. Which of the following bond is formed as a result of reaction of carboxylic group of one amino acid with amino group of other amino acid with the elimination of water? Tell me, which type of bond do you find between amino acids? That's all is the question. Question is nothing but which type of bond do you observe between the amino acids? That is, when many amino acids join together, they make up the proteins. Which amino acid is, which type of bond is between proteins? That is peptide bond. That is going to be peptide bond. Done. Now students, there are different functions of proteins. Different functions of proteins: structural, contractile, nutrient, regulation, defense mechanism, transport. Some of the proteins can also show some type of catalytic activity. Yes, some of the proteins can show certain catalytic activity. They can show, and that is what we'll be talking right now. That is catalytic property of protein, and that is nothing but we call that catalyst. That's what we called catalyst. Let me know in the chat if you're ready for the next concept for today. That is going to be catalyst now. Next concept for today, catalyst. That is, catalysts are basically type of proteins. Catalysts are basically type of proteins. Not catalyst, I'm talking about enzymes. Enzymes are proteins, but catalyst, that is broadly speaking, catalysts are, in chemistry they won't be proteins obviously. In biology they'll be proteins. Okay. Now let me know in the chat if we can start the next concept for today. The next concept for today is going to be the catalyst. Now we are coming towards the difficult part. Do you realize carbohydrate is done, dusted. Then we did the structure of proteins, done. Now this is where many students face difficulties. They do not understand the different properties of catalyst or the different mechanisms of catalyst. What is this activation energy, and what is this Km value, what is the substrate concentration, that is what is Vmax, all that will be starting now. What is this inhibitors, competitive, uncompetitive, non-competitive, what is this cofactor, and what is this coenzyme, apoenzyme, holoenzyme. Do you also have confusion in following concepts? Tell me yes or no in the chat. Do you also have confusion between what is apoenzyme, what is holoenzyme, right? What is competitive, uncompetitive, and non-competitive inhibitors? Yes or no? Do you have confusion between them? If yes, all the confusion will be clear today. All the confusion will be cleared in today's video, and you don't have to worry. Everything is going to end today. Everything's going to end today. Biomolecules chapter, the fear you have for biomolecules chapter is going to disappear. And trust me students, trust me when I say this, when you sit and solve questions, after today's class of biomolecules anywhere, you will be able to solve all the questions if it is from NCERT. If it is from NCERT, I guarantee you, you will be able to solve every single question from biomolecules, however difficult it is. Because I'll make sure you get a deeper understanding of the concept and also the NCERT lines. Okay? So can we start now? What is a pineapple? It's not a it's a fern. Nami is adding a fern, is it? I thought it was a pineapple. It is a fern. Okay, ferns are nice. Ferns are beautiful. Okay. Let's understand catalyst, students. Now in terms of chemistry, what is a catalyst? Catalyst is a type of, let me write it down. It is a type of substance. It is a type of substance which basically alters the rate of reaction. Rate of a reaction. Alters the rate of a reaction. It can increase the rate of reaction or it can even decrease the rate of reaction. Yes, you can increase the rate of reaction or decrease the rate of reaction without getting consumed. Without getting consumed. Yes, without getting consumed, a catalyst can alter the rate of reaction, increase the rate of reaction or decrease the rate of reaction. Concept done. Concept is done. Now, there are two types of catalyst. Two types of catalyst. One is inorganic catalyst, which we learn in chemistry. Example is platinum. Then we have organic catalyst. And we call the organic catalyst or the catalyst found in the living tissues, we call that enzymes. We call them enzymes. Simple example is trypsin. Okay, we have a lot of enzymes. In glycolysis you learned many enzymes. You learned in redox, you learned many enzymes in the case of your Krebs cycle, right? In the case of digestion chapter, you might have learned about so many enzymes. So all these enzymes are basically type of catalyst. Now, let's understand into a deeper version of what is the enzyme now. What is an enzyme? First of all, let me tell you, all enzymes are made up of proteins. They're made up of proteins except ribozyme. Ribozymes are basically RNA enzymes. They are RNA. They are RNA enzymes. So all enzymes are typically made up of proteins and except the ribozyme, which is a type of RNA enzyme, which is a type of RNA enzyme. Now all of the enzymes are extremely highly, they are very highly specific in action. They are highly specific in action. One enzyme will have only one type of substrate. It will join to only one type of substrate. They are very choosy, very picky. They are very picky, very highly in enzyme. Their major of the work is in aqueous medium. They need a medium to work. In aqueous medium, their main function works. All the enzymes in the cytoplasm, or it could be in your Golgi bodies, or it could be in your mitochondria. Aqueous medium is required for functioning of enzymes. Now, if everyone has a small understanding of enzymes now, yes, small understanding of enzymes is there. Now, what is this active site then? What is this active site? Okay, students, when we were learning tertiary structure of protein, this is woolen ball structure of tertiary structure of protein. In this tertiary structure of protein, students, there can be certain pockets. Or crevices in the tertiary structure of protein, students. In the tertiary structure of proteins, there can be certain pockets or crevices. Can you see? This right here is a pocket. This right here, crevices or pocket. This right here. This crevices or pocket will have certain types of functional groups. Certain types of functional group will be there. Functional groups will be there. It is R. Such functional group which is found in a pocket or a crevices of the structure of a protein. On the structure of a protein, that is called as active site. This is called as active site. And active site is the point. Active site is the point in the protein where the substrate will come and attach. The substrate will diffuse. And come and attach at this point right here, and that is called as active site. This is the structure of protein. On proteins, there will be active site. Obviously enzyme. Obviously it's an enzyme only. Protein is an enzyme. Okay. So if you look at this DNA polymerase, this is the structure of DNA polymerase. On this DNA polymerase, there are certain pockets or gaps. In this pocket or gap, we have something called as polymerase active site. We have the polymerase active site is present. That is a small crevices or a pocket in which functional groups are present. Is the point clear to all of you about active site now? All of you understood what is the active site? Yes. Active site is done. Okay. So read this. All enzymes are proteins except the ribozyme. Okay. All enzymes, like any protein, has a primary structure. Amino acid sequence of the protein. Enzyme, like any other protein, has a tertiary and quaternary structure, secondary and quaternary structure. When you look at the tertiary structure, you will notice the backbone of the protein chain folds upon itself. Backbone, beta plated and helical, they will interfold. In the tertiary structure of protein, you will notice many crevices or pockets are made. We drew the structure. Small crevices or pockets are made in the tertiary structure of protein. Yes. One such pocket is called as active site. See here. It's called as active site. Done. All of you learned it today. An active site is an active site of an enzyme is crevices or pocket into which substrate fits. So substrate will diffuse and come and it will fix on the active site. Done. Active site. Thus enzyme, though their active sites catalyze the reaction at high rates. So enzymes are able to function the high rates because of the attachment of substrate at the point of attachment is called as active site. Now students, every single enzyme will work at a specific temperature. Inorganic enzymes, inorganic catalysts will easily work at higher temperatures. But organic enzymes, organic catalysts, enzymes can usually not work at high temperatures because they denature. See, usually if the temperature increases, what happens? I told you right now that all the enzymes are made up of proteins. And if the temperature is very high, what happens? All the proteins will denature. They will denature. Yes, they will denature. But there is this one enzyme which does not denature, which is obtained from a Thermus aquaticus, an organism which survives in a very acidic environment, very high temperatures, hot springs, Thermus aquaticus. And the enzyme which is produced from Thermus aquaticus is called as Taq polymerase. It is called as Taq polymerase. See, a thermal stable is an important quality of an enzyme isolated from thermophilic organism. That is Thermus aquaticus, and you will study about that organism in the PCR cycle. In the PCR cycle you will understand. PCR cycle you will understand. Now students, we have a simple reaction here. CO2 plus water, carbonic acid. Without catalyst, let me put a perspective how important an enzyme is. How important an enzyme is actually is for a particular reaction. Perspective. Understand. Understand the perspective. For example, we have CO2 here, water and carbonic acid. Usually there are 200 molecules are produced. 200 molecules are produced in 1 hour. 1 hour. 200 molecules in 1 hour of carbonic acid are produced in 1 hour. But if you add an enzyme, that is carbonic anhydrase, if you add an enzyme carbonic anhydrase, then 6 lakh molecules is formed in 1 hour. Same 1 hour, 10 minutes or 1 minute. No, not in 1 minute students, in 1 second. In 1 second. In 1 second, 6 lakh molecules will be formed. 1 second. That is technically around 100 million times faster. 10 million, sorry, not 100. 10 million times faster. 10 million times faster when we compare it to without catalyst. When we compare it to without catalyst. That is the incredible power of enzymes. Incredible power of enzymes, students. That is the difference. That is the difference. That is with enzyme, without enzyme. With enzyme, without enzyme. See here. Example is given, same example is given here. Same example is given here. Now here you need to understand the rate of reaction. They will never ask you this in general. But it's very easy. That is, rate of physical or chemical process refers to the amount of product. Amount of product per unit time. Per unit time is called as rate of reaction. If you add a direction to it, we can call it velocity. Velocity if direction is specified. Okay. They will never ask you all this. It's very easy things. It is influenced by temperature among the other following. Thumb rule is what? If you increase the temperature, by 10 degrees Celsius, the reaction will be faster. If you decrease the temperature by 10 degrees Celsius, the reaction will be very slow. That's all. It will be decreased by half. It's given here. See, very slow reaction. 200 molecules of carbonic acid is found in 1 hour, but with catalyst 6 lakh molecules is being formed in every second. 6 lakh molecules is formed every second. Okay, see this line. There are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction when each of the step is catalyzed by a same enzyme or complex or different enzyme. For example, in glycolysis, multi-step process. In multi-step process, every step we have a different enzyme. When it comes to glycolysis, when it comes to Krebs cycle, every single step we have a different enzyme, isomerase, hydrolase, and everything, right? So multi-step process. See, glucose to pyruvic acid, multi-step process. Now students, Now we come to classification of enzymes. Now we come to the classification of enzymes. Now I'll be teaching you what is apoenzyme, what is going to be your holoenzyme and everything. Okay. Can we start? Friends, can we take a 5 minutes break and we can start again? Because I want 100% attention now. Now I'll be teaching you what is apoenzyme, what is cofactors and everything. We will take a simple 5 minutes break and we will start again. What's it? That is now, the time is 1:15. At 1:20 we'll start again. Okay? 1:20 will start. 1:20 will class resume. 1:20 will resume the class. Okay? Class will resume at 1:20. So after that because I want 100% attention now. Because the next topics which I'm going to teach after the break, right? The next topics are going to be so important for you. You will realize that many questions will come from this enzyme part. Many questions will come from so many different factors here. Okay? Let's take a 5 minutes break and we'll come back. Let's take a 10 minutes break, students. 1:25 will start. 1:25. We'll start at 1:25. 1:25 we can start. 1:25. So all of you can relax a little and then we can start. 1:25 we can start. Okay? 1:25 we can start. So let me know, chat, we can start the class again, we can resume the class. All of you went away, I feel. Let's resume the class 1 second. Yes. So students, now is going to be the most important part. I want everyone to concentrate, like, double extra now, double triple extra concentration, ultimate concentration. Okay, because if you miss even a 1 point, there's a very high chance that you might not understand the concept at all. So I want everyone to completely focus right now. Now students, let's understand by something called as simple enzymes. Simple enzymes consist of only amino acid, only the protein part. Simple enzymes are like we normal humans. Simple enzymes are like your normal humans. Normal humans. We are the simple enzymes. We are made up of protein. We are made up of protein. Imagine, we are made up of proteins. Okay. Now we have something called as conjugate enzymes. Conjugate enzymes. The conjugate enzymes have two different parts. Remember students, conjugate humans are like your superheroes. Superheroes. Let's take a quick example. Let's take a quick example of a superhero, that is, let's say Flash. Let's take Flash for example. Okay, let's take Flash for example. The Flash, before he got the superpowers, was he also human? Yes. Before the Flash got his superpowers, he also was like a normal human. That is, only protein part was there. Yes, only protein part was there, and we call the only protein part as apoenzyme. Apoenzyme is called as the protein part. That is the Flash when before he became superhero. That is Barry Allen before he became a superhero. Okay. Now, once he got the powers, once the Flash got the powers, the power is called as what? Cofactor. The power is called as cofactor. It is the non-protein part. And do this non-protein part of the cofactor, is it present in simple enzymes? No, it is not present in simple enzymes. It is only present in conjugate enzymes. And students, when there is only protein part, the Flash was human, so biologically inactive. It means he was not able to save people. He did not have superpowers. He was not able to run very fast. So it is biologically inactive. Only protein part, apoenzyme. But when the powers came into the picture, that is, when the cofactor is added to the apoenzyme, when cofactor is added to the apoenzyme, that is called as holoenzyme. So what is holoenzyme? Holoenzyme is nothing but apoenzyme plus cofactor. And remember, only apoenzyme is inactive. Only protein part is inactive. But when you add the cofactor, it activates the catalytic activity. The power, this is the power, right? This is the power. The power is given to the normal Barry Allen, and he becomes a Flash. So what do we learn now? We learned that we have apoenzyme, which is the protein part, which is biologically inactive. Then we have cofactors, which are non-protein part. When only apoenzyme is present, it is inactive, biologically inactive. But once cofactor is added, to the apoenzyme, together they are called as holoenzyme. And remember students, only the holoenzymes are complete and biologically active. They're biologically active. Why? Because of cofactor. Now imagine cofactor is not present. What will happen? Tell me in the chat. If cofactor is not present, then the holoenzyme will not be active. It will be just remain as apoenzyme. Holoenzyme will not be active at all. Is that concept clear to all of you? Tell me in the chat, yes or no. That is, holoenzyme is apoenzyme plus cofactor. If only apoenzyme is there, it is biologically inactive. when we add cofactor to it, it becomes biologically active. Okay. Now what is cofactor? What is this power which we are talking about? Cofactors are actually of two types. Cofactors are actually of two types. We can have them as metal ions, we can have them as simple metal ions, or we can have them in the form of, I think there are three types. Metal ions, they can be coenzymes. Let me write here. They can be prosthetic groups, or they can organic. Organic can be divided into prosthetic groups, or they can be divided into coenzyme or coenzyme. So there are three types of cofactors. There are three types of cofactors. So simple enzyme, what is the reaction they will do? Simple enzymes are, they do not have much of functions, they will aid in the other functions, that's all. Okay. So metal ions, they have metal ions, the first one. Second one is prosthetic groups, and third one is coenzyme. Three different types of cofactors are present. All of you know what is the importance of cofactor? Cofactors biologically activates the apoenzyme. Now, first let's understand about metal ions. Simple example which is given you in NCERT. Simple given example in NCERT is your zinc. Zinc is a cofactor. Zinc is a cofactor which is present in the carbonic anhydrase. It is present in carbonic or carboxypeptidase. One more is carboxyl peptidase. This carboxyl peptidase, students, carboxyl peptidase or carboxyl anhydrase, they are the apoenzymes. The cofactor here is the zinc. Zinc will activate the carbonic peptidase or carbonic anhydrase. If zinc is absent, there is no functioning happening at all. That is the importance of zinc here. Okay. The other one is organic. In organic, there are two types which I will tell you. In organic, there are two types. One is prosthetic group, other one is coenzyme. Prosthetic are, can I say, tightly bound. They are very tightly bound, while your coenzymes are loosely bound. They are very loosely bound here. Coenzymes, they are very loosely bound. Example for prosthetic is hem group, and for coenzyme, which we'll be discussing later on, is going to be your example is NAD plus or NADPH or NADH. Let's take it as NADH, not DP. NADH. These are the examples. For example, students, let's go to one by one. Let's you understand it better. Your NADH is the coenzyme which activates the lactate dehydrogenase. It activates the lactate dehydrogenase. So in the lactate dehydrogenase, we have the NAD+. If NADH, the coenzyme is not present, coenzyme is a type of cofactor. If NADH is not present, the enzyme is useless. So NADH is the coenzyme, which is a type of cofactor. And remember, students, your NADH is made up of a vitamin, that is niacin vitamin. The structure of NAD+ will have niacin along with it. Okay. Now other one was coenzyme is tightly bound. We discussed this. Prosthetic group example is going to be peroxidase and oxidase. Peroxidase and oxidase are the apoenzymes. This peroxidase or oxidase, they have the heme group. In the middle. If you look in the middle, they have the heme group. HAEM. If heme group is absent in peroxidase, the function will not happen. So heme is basically the prosthetic group here, which is a type of cofactor, which is a type of cofactor. If heme group is absent, peroxidase is not functioning. What is the meaning of peroxidase? The main function of peroxidase is to catalyze the hydrogen peroxide reaction. Hydrogen peroxide reaction catalyzing. See here, students, read the lines of NCERT, you will understand better. See here, cofactor. Enzymes are composed of one or more several peptide chains. However, there are a number of cases in which non-proteinaceous content are called as your cofactors. Cofactors are non-protein part. Okay. Now, they are bound to the enzyme, that is, they are bound to the apoenzyme to make the enzyme catalytically active. They are bind to the apoenzyme and they're making it a hollow enzyme and they're making it catalytically active. In the instance, the protein part of the enzyme is called as apoenzyme. Okay, enzyme. So apoenzyme plus cofactor, hollow enzyme. Okay, three kinds of cofactors. One is called as your prosthetic group. In prosthetic group, we just now learned they are very tightly bound. They are very tightly bound to the apoenzyme. Very tightly bound to the apoenzyme. Example is peroxidase and oxidase are the apoenzymes. They are the apoenzymes. Okay, which catalyze the breakdown of hydrogen peroxide to water and oxygen. That is their function. This is the apoenzyme. Now, which is the prosthetic group here? The prosthetic group here is going to be heme. Heme is the prosthetic group. And without heme, this is not going to work. The function is not going to work. So heme is the cofactor or prosthetic group which makes the hydrogen peroxide catalytically active. Without heme, peroxidase is not going to work. Peroxidase and catalase is not going to work. Heme is the prosthetic group and it is part of the active side of the enzyme. That is the first one, prosthetic group. Second one is coenzyme. We learned it, coenzyme, right? First of all, they are loosely bound. They are very, association is very, with the enzyme is very transient, that is very loosely bound here, very loosely bound. Coenzymes serve as cofactor in number of different enzymes catalyze reaction. Example is very here. Coenzyme nicotinamide adenine dinucleotide and NAD+. They are the coenzymes here. They are the coenzymes which make other apoenzyme activate. And they also contain vitamin niacin. So NAD+ and NADP are the cofactors. They are the cofactors. Remember, don't get confused. They are the cofactors here or coenzymes. Okay. The last one is going to be metal type, metal ions. Last one is the metal ions, which are inorganic type of it, inorganic. Their activity which forms the coordination bonds with the side chain of the activity site at the same time. Okay, this is not, example is very important. Example, see, zinc is the cofactor for the proteolytic enzyme carboxypeptidase. Carboxypeptidase is the apoenzyme. Without zinc, carboxypeptidase is not going to work. So once we have the carboxypeptidase apoenzyme, zinc is coming, making it a hollow enzyme, and it will be catalytically active. Carboxypeptidase will be catalytically active then. Catalytic activity is lost when the cofactor is removed. Told you, once you remove the cofactor, once you remove zinc, once you remove the NAD+, or once you remove the heme part, the function is gone from the enzyme, which testifies that they play a very crucial role in the catalytic activity of the enzyme. Catalytic activity of the enzyme. Are you understanding the concept, students? Concept is very important here. That is the concept. See this question now. Okay, this we have to study later on. This I have not taught you still. We have not done that yet. See this question now. The cofactor of enzyme carboxypeptidase, which is the cofactor of carboxypeptidase. Without this cofactor, they will not function. They will not function. The answer is going to be zinc. Without zinc, carboxypeptidase will not work. Carboxypeptidase will not work. Right? And where do you get heme? Where do you get heme, students? Heme is a prosthetic group. Heme, will find it here. Heme. Heme in the case of peroxidase and catalase. Heme in the case of peroxidase and catalase. And zinc in the case of, where did zinc go? zinc in the case of carboxypeptidase. Answer, everything needs to be done with the help of examples here. Now students, now we learnt about cofactor. We learnt about cofactor, we learnt about coenzyme, we learnt about a prosthetic group, we also learnt about apoenzyme, holoenzyme, everything is done. The next concept which is going to be important right now, the next concept is going to be how do the enzymes do it? Like how are they able to do it? How are they able to catalyze the reactions? What is their secret behind it? It's very simple. It's very simple. That is, first they have reactant 1, reactant 2, also called it substrate. They make some, then the product formation is happening. What is happening in chemical reaction? Formation of breaking of bonds is happening. Okay. Now, there is something called as reactant plus reactant. When the reactants are reacting, they make an intermediate product, also called as a transition state. And this transition state right here has very high energy. And then we finally get the product from there. Okay, the transition state is there. Intermediate product is formed, which has very high energy. Now, you need to understand, if you want to learn the function of enzymes, how are they doing this, how are they catalyzing the reaction, there is a concept called as activation energy. If you understand the concept of activation energy, everything will be very clear to you now. Everything will be very clear to you. To understand the concept of activation energy, it is the average energy difference between, average difference between substrates, the reactants, substrate or reactants and the transition state. That is called as activation energy. If you go back here, I told you the reactants will have some energy, the transition state will have some energy. The energy difference between reactants and the transition state, that is called as activation energy. The energy difference between reactants as well as the intermediate product, that is called as activation energy. Now if you look at here, this is the reactant, this is the product. This right here, can you see the top peak? This is the peak here, this is the activation energy. Now this is the energy of the, this is the activation energy required. So we have reactant here, we have the peak here, that is the, one second, the GIF to start, one second, let me go back. Here we have something, this is where we get the transition state. Okay, here we get the transition state. Now here there is energy, there is an energy here. Okay, this I told you, this is called as activation energy. The difference in energy between reactants and the transition state, this is called as the activation energy. Now what is enzymes doing? Enzymes are reducing the activation energy. Can you see? This is the top peak. Now enzyme will do what? Once you add the enzyme, the activation energy will be decreased. That is the function of enzymes. So imagine if you're trying to do high jump, you're trying to do the high jump. Now imagine the bar is still here. Will you be able to jump easily? No. It will be very difficult. Imagine if someone is coming and they're reducing the bar to down. That is, they're keeping the bar very low. Then you can easily jump over. Yes. So what is your enzymes are doing? Enzymes are basically reducing the activation energy. They're reducing the activation energy. If activation energy is reduced, reactants can easily make the products. Reactants can easily make up the products. Done? Done? Tell me students, is it point clear to all of you? That is, the main function of enzyme is to reduce the activation energy. What is activation energy? Activation energy is the average energy difference between reactants and the transition state. Here, this is the activation energy, and your enzymes are basically making it reduced. The energy difference they're reducing. That is the function of your enzymes. So we have substrates, enzyme, substrate complex, transition state structure. Complex is formed. Now eventually the complex will be broken down and we get the enzyme product complex. Then we finally have the product plus enzyme is separated out. I will repeat again. First we have the substrate plus enzyme substrate complex. Then we have enzyme product complex, and then enzyme is coming out, product is coming out. Product is sufficient, and we always tell, right, enzyme is not consumed in the reaction. Enzyme is not consumed in the entire reaction. Done. That is the mechanism of action. Now in the chemistry or physics you might have learned, right, always you have learned there's something called as endothermic reaction and exothermic reaction. What happens in endothermic reaction? Tell me in the chat. What happens in endothermic reaction? In endothermic reaction, energy is consumed. In exothermic reaction, energy is released. But who is consuming, who is releasing, students? Who is doing it? Understand the concept now. See here. In the case of your endothermic reaction, in the case of your endothermic reactions, reactants are absorbing the heat. Reactants are absorbing the heat and they're able to cross activation energy very well. That is why it's called as endothermic reaction. In endothermic reaction, reactants are absorbing the heat and they're able to cross the activation energy very easily. What about in exothermic reaction? In the case of exothermic reactions, the reactants are losing the heat. Reactants are losing the heat and they're forming the products here. They're forming the products. That is the difference between endothermic and exothermic. In exothermic, reactants are losing the heat, product formation. In the case of endothermic, reactants are absorbing the heat and they're making the products. Simple, right? Understood? That is the difference between endothermic as well as exothermic. This is a diagram given you in NCERT. Do you see this? Activation energy without enzyme, very high state is there, transient state. What is activation energy? The energy difference between substrate and the transition state. This is the activation energy. Enzyme is reducing the activation energy. So can you see the pink line? The pink line is with enzyme. Reduced activation energy. So substrate can easily make the products. Substrates can easily become the product right now. That is activation with the enzyme. That's what we learned right now. See here. Enzyme substrate complex, enzyme product complex, enzyme is separate, product is going to be separate. See, first the substrate binds to the active site. Remember always, substrate is diffusing and coming. Substrate is diffusing and coming and it is binding with the active site of the enzyme and fitting into the site. The binding of substrate induces the enzyme to alter its shape more tightly around the substrate. So it will completely bind. So enzyme is completely binding with the substrate now. The active site of the enzyme now is in close proximity of the substrate, breaks the chemical bond of the substrate and new enzyme product complex is formed, EP. The enzyme released from the product of the reaction and the free enzyme is ready to bind to the one more later on. Look at this question. The question says that in a reaction catalyzed by the enzyme, which of the following statement is correct? Okay, read the options. Enzyme decreases the activation energy for formation of transition state. Enzyme makes the transition from substrate to produce more product. Enzyme increases the activation energy. Enzyme substrate complex formed during the reaction lasts for very long. No, this is the answer. To form the transition state, there is decreasing the activation energy. Look at this question from NEET 2024. NEET 2024 question. Regarding the catalytic activity of enzymes, select the correct sequence. Select the correct sequence. First, what happens? Substrate binds to the active site. Yes. substrate enzyme complex. Okay. Then chemical bonds are broken down. Yes. Then release of a product is released. Then free enzyme is ready to bind to the another site. So one is going to be EA EADCB here. 4 is the answer. 4 is the answer. Yes. Now, there are many factors which affect the activity of enzymes. Temperature, pH, if there is no optimum temperature, if there is no optimum pH, if there is no proper substrate concentration, enzyme concentration, inhibitors. All of this play a very crucial role in the activity of the enzyme. For example, temperature. If there is a very high temperature, enzyme will denature. If there is a very low temperature, the enzyme will completely become frozen, they become inactive. So there is something called as optimum temperature is there. Denature, inactive. So they need a very optimum temperature at which all the enzymes will work. Okay. Similarly, there is something called as optimum pH. For example, pepsin it is around 1. For salivary amide it is at 6. For alkaline phosphate it is around 9. That is called as optimum pH. At that optimum pH, a particular enzyme is going to work very well. Okay. Optimum pH. Now, what about substrate concentration? What is this substrate concentration, friends? Listen to me very carefully. Here we have at the bottom, let's keep it as substrate concentration. X-axis is substrate concentration. Y-axis, let's keep it as reaction rate. Now, initially we have the enzymes are free. Yes, enzymes are going to be completely free. Some of the enzymes are attached to the substrate. This round is the substrate. This is the enzyme. This U shape is going to be, this pocket is going to be active site. Now, what are we doing? We are increasing the concentration. We are increasing the concentration of substrate. As we increase the concentration of substrate, the rate of reaction is also going, obviously. More substrate, more enzymes, reaction is happening faster. Rate of reaction is going in medium substrate. What about in very high substrate reaction? When the substrate is very high, is there any increase of reaction? No. That is, at a certain point, the reaction can become very constant now. Reaction will become very constant now. So, how much ever you increase the substrate, the rate of reaction is not going to increase, because all the enzymes are already occupied. Okay, already occupied. Now, we get to the concept of something called as Vmax or half Vmax. What is this Vmax and what is this half Vmax, students? Vmax is basically, Vmax is nothing but maximum velocity at which all active sites are filled. Maximum velocity at which all active sites are completely consumed. Okay, can you see? Probably this is the Vmax right here. At this point, all the active sites are completely filled by substrate at this point. But students, Vmax is very difficult to measure. It is very difficult to measure. So what do? We make something called as half Vmax. It is basically the maximum velocity at which half of the active sites are completely filled. Half of the active sites are filled. Half of the active sites are completely filled here. Half of the active sites are completely filled. That is half Vmax. Now, if this is complete Vmax, this is the half Vmax. Now, at this half Vmax, I told you, half of the active sites are filled. It means that to fill that half of the active sites, some amount of substrate should be there. This is the active site. This is the active site here. Okay. At this active sites, some amount of substrate is being added here. Yes, some amount of substrate is being added. The concentration, students, listen to me very carefully. The concentration of substrate at half Vmax, the concentration of substrate at half Vmax is called as Km value. That is Km value. Km value, Michaelis constant. Km value is nothing but concentration of substrate at half Vmax. At half Vmax. Concentration of substrate at half Vmax is called as Km value. It is called as Km value. Can you see? Here is the Km value right here. It is half Vmax is here, Km value is here. Km value is there. Now, I'll give you a simple question. Okay? We have two reactions happening. Two reactions are happening here. Why this curve? Because as we increase the substrate concentration, rate of reaction also increase. Rate of reaction. Substrate concentration is here, and this is rate of reaction. This is rate of reaction. As we increase the substrate, rate also is increasing. Okay, rate is also increasing. Now, for example, this is the Vmax here. This is also the Vmax here. Vmax. For reaction A, half Vmax is here. Half Vmax. And this will be Km value. This right here will be Km value here. Km value. For example, this is 0, this is 1, this is 2 and everything. For the reaction B, reaction A and reaction B. For reaction B, this right here is the half Vmax value. Half Vmax value. Okay. So this right here is going to be the Km value. Km value. Km value. Now, this is again 0, 1, 2, 3. Now, if I ask you a question, which of the following reaction, which enzyme in reaction A or reaction B, which enzyme is better? Which enzyme is better? Let me ask you, which enzyme is better? You will say option number B or option number A. Which enzyme is better? Remember always, a good enzyme will have low Km value. It will always have a low Km value. What is the meaning of low Km value, students? The meaning of low Km value is that it is requiring very less number of substrate. It requires very less number of substrate to reach the half Vmax. So can you see this option number, reaction A? Reaction A is able to reach half Vmax value at a very low substrate. If you add even a little bit of substrate, it is reaching half Vmax. But reaction B, students, reaction B requires a higher Km value. It requires more substrate. To reach the same half Vmax. To reach the half Vmax. So obviously reaction A is better. The enzyme reaction A is better. Are you understanding? That is the concept of Vmax. Now, students, next thing you need to understand is enzyme concentration. Okay, enzyme concentration. Enzyme concentration is always just like your substrate concentration. It is just like the substrate concentration. Here we have rate of reaction. Here we have enzyme concentration. Obviously, students, enzyme concentration will eventually, starting it will increase with the increase in the enzymes. Eventually, again, it will become stagnant. It will become constant, just like substrate. This graph is just like substrate concentration. Initially the reaction will increase because increase in enzymes, but later on, once it becomes saturated, once there is no more substrate available, reaction will become very constant later on. Reaction will become very constant later on. Constant reaction will happen. Okay. Now the last part of the concept, the second last part of today's concept is going to be inhibitors. Inhibitors. What are inhibitors? What are the different types of inhibitors? Are you ready to learn now? Ready? Tell me in the chat quickly. Are you ready to learn the last part of today's class? That is going to be the inhibitors. Let me know in the chat if you're ready for the last part. Ready? I hope there was no doubt, right? I hope that entire Km value, again we'll come back to that. It was easy to understand, right? It was very easy to understand. There's no difficulty in that, right? Now, what is inhibitors? Inhibitors basically are chemicals that shut off the enzyme activity. It is like, calm down bro, calm down. No more activity for you. Your activity is going to completely stop. Your activity is going to completely stop now. It is stopping the activity. In other words, inhibitors inhibit the enzyme activity. But how do they do it? How do they inhibit the activity? That is more important to understand. How are they inhibiting the activity? Okay, once again students. How are they inhibiting the activity? How are they inhibiting the activity? Now, there are three types of it. There is competitive, non-competitive and uncompetitive. Three different types of inhibitors. Each of them will function very differently. For example, all the competitive inhibitors, they are in a competition. All the competitive inhibitors will compete for the same active site. Compete for the same active site. For example, look at this. For example, both of them look very alike, but this red one is a inhibitor. This is your substrate. Green one is the substrate here. Substrate. Instead of substrate joining to the active site, inhibitor will join into substrate site and become inhibitor enzyme complex. Enzyme inhibitor complex will form. So it is directly competing. It is directly competing for the active site, and inhibitor will look just like the substrate. See here. Resembles the substrate, compete with the substrate for the active site. So substrate will not join, Papa. Your inhibitor will go and join, and the reaction will not happen. Reaction will not happen. Now students, this is important to understand. This is important to understand now. Now, in a reaction, in an entire reaction, if the substrate is not able to join with the enzyme, if the substrate is not able to join with the enzyme, will there be increase in substrate level concentration or decrease in substrate concentration in the reaction? Tell me. In an entire reaction, your substrate is not able to join with the enzyme. So obviously in the reaction, substrate concentration will be more left out, right? So can I tell the Km value? The Km value will obviously increase. Km value will increase in the reaction. Km value will go on increasing in the reaction because it is not able to join. It is not able to join there. Okay. But even though the substrate is not able to join, what will happen with the rate of the reaction? Some of them will be joined, some of them will be joining, but the final rate of the reaction, students, the final rate of the reaction will always remain the same. Unless there is a major change, Vmax will remain the same. Vmax, maximum velocity. Vmax, maximum velocity at which all the active sites are filled, will remain the same, because after some time, all the active sites will fill up. All the active sites are filling up, right? All the active sites are filling up. So Vmax is going to remain the same because all the active sites are filling up. That is the Vmax, velocity at which all the active sites are filling up. Sometimes enzyme is filling, sometimes inhibitor is filling. So sometimes substrate is filling up, sometimes inhibitor is filling. Okay, so Km value will increase in the reactants. Km value will completely increase. The example is very important. Example is very important. Not given here. Example is going to be succinate to fumarate. Succinate to fumarate. The enzyme involved here is succinate dehydrogenase. Succinate dehydrogenate enzyme is there. Now we have one more inhibitor called as malonate. We have an inhibitor called as malonate. This malonate looks just like succinate dehydrogenase. So this malonate will completely go and replace the succinate dehydrogenase. It will completely replace it will go and bind with the succinate dehydrogenase enzyme. Okay? So fumarate will not be formed. Fumarate will not be formed. Now if you look at the non-competitive is very simple again. It binds at a site other than the active site, not active site, other than the active site, somewhere else it is binding. Okay? It changes the shape of the active site such that the substrate is not able to bind. See, can you see? It will bind. Changing of the active site will happen. Now this substrate is not able to bind at all. That is called as non-competitive. Non-competitive is very simple. Non-competitive inhibitor will join other than the active site and it will completely change the shape of active site and your substrate will not be able to join. It can bind to free enzyme also. It can bind to free enzyme or it can also bind to the enzyme substrate complex. Even though complex is formed, it can still come and bind. It can come and bind still. Done. That is non-competitive. In non-competitive is very simple. In non-competitive, your entire reaction only is changing. Vmax is reducing here. Complete Vmax is reducing. Substrate level is going to remain the same. Km value will remain the same. Km will remain the same. Vmax will decrease here. Competitive inhibitors and non-competitive inhibitors. Uncompetitive inhibitors. Now what about, this is non-competitive, right? What about uncompetitive? They don't want to compete in the starting. They don't want to compete in the starting at all. That is, this uncompetitive enzymes will wait. They will wait until the complex is formed. See here, until enzyme substrate complex is formed, they will wait until then. They will not join the starting. Once it is joined, then it will join and it will kick it out. So uncompetitive, they will not compete in the starting. They will wait for competition to happen. They will wait until the complex is formed. Once the complex is formed, then they will join and then they'll kick out the substrate. So it binds to the enzyme at the site other than the active site. It cannot bind to the free enzyme. It will not bind to the free enzyme. It will only bind to enzyme substrate complex. Only bind to enzyme substrate complex. That's all. Let's see the NCERT lines. See here, temperature and pH we learned. There's something called as optimum temperature and there is something called as optimum pH. Optimum temperature and optimum pH. High temperature destroys the enzymatic activity. Protein gets completely denatured. Low temperature it becomes inactive. Concentrated substrate I told you. With the increase in substrate concentration, the velocity of the enzymatic reaction at first, the velocity of enzymatic reaction rises at first. Starting it will increase properly. Okay. The reaction ultimately reaches a maximum velocity that is called as Vmax, which is not exceeded by any further raise in concentration, maximum velocity. This is because the enzyme molecules are fewer than the substrate molecules. And after the saturation of these molecules, there are no more free enzyme molecules to bind an additional substrate. See, basically at the one point, when you how much of an enzyme you include, will there be no substrate left. That is the concentration of substrate. Or if there is no more enzyme left for it to bind. How much of a substrate you increase, there will be no more enzymes to bind, so reaction becomes constant. Enzyme molecules are very fewer, so enzymes are very fewer compared to substrate. Then we have inhibitors, which completely stop the reaction. Competitive inhibitors, structurally similar with the substrate, compete with the substrate for the substrate binding site. Okay, example is inhibition of succinate dehydrogenase by malonate, which closely resembles the substrate succinate. So malonate is resembling the substrate, so it is not allowing the substrate, that is succinate, to bind at all. So if you go back, your malonate will completely bind with succinate dehydrogenase. It will not allow succinate to bind at all. It will not allow succinate to bind at all. Malonate will completely bind with succinate dehydrogenase. And students, such competitive inhibitors are often used in bacterial pathogen studies. Bacterial pathogenic studies. Optimum temperature, optimum pH. This is the half Vmax. This is the Km value here. Km value. See this question. Which of the following statements regarding enzyme inhibition is correct? Enzyme inhibition. Competitive inhibitors is seen when substrate competes with the enzyme for the binding to an inhibitor protein. Competitive inhibitors is seen when the substrate competes with the enzyme. No. Competitive inhibitors is seen when the substrate and the inhibitor compete for the active site. See, both are competing with the active site for the enzyme. Non-competitive inhibitors of an enzyme can overcome by adding a large amount of substrate. No. Non-competitive inhibitors often bind to the enzyme irreversibly. No, it is reversible. Answer. Substrate and inhibitor both will fight for the place in the enzyme, for the location in the active site of the enzyme. Malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of succinate dehydrogenase. Its binding activities decrease completely. Low temperature preserves the enzymes in a temporary inactive state. High temperatures destroys the enzymatic activity because of the protein are denatured by heat. Yes. When inhibitor closely resembles the substrate in a molecular structure and inhibits the activity of the enzyme, it is called as competitive inhibitor. Yes. Both are true. We just now learned the entire structure. Now students, with that being said, we are towards the end of the chapter now. I believe all the concepts are getting clear. The only way you will feel more confident in this chapter when you solve a lot of questions. Now unfortunately I don't have a lot of questions right now. I have few PYQs, but if you want to solve more questions, you know where to go. The NEET PDI is there, right? NEET PDI is there, and we have been giving you a lot of questions, so everything is there in the treasure box. So solve more questions, solve more PYQs, you'll become more and more confident. Now we go towards the last part of the chapter, that is the different types of enzymes. Based on the chemical reaction, we have oxidoreductase, transferase, hydrolase, lyase, isomerase and ligase. Different types of enzymes are there. For example, first one is oxidoreductase. We have the oxidoreductase, where simple example is pyruvate. Pyruvate is getting converted to acetyl-CoA in the link reaction. Remember, in the link reaction, pyruvate is getting converted to acetyl-CoA. Enzyme is going to be oxidoreductase enzyme. Oxidoreductase enzyme. Here it is a dehydrogenase enzyme. Dehydrogenase enzyme is there. Dehydrogenase enzyme is there. Now, what about transferase? Transferase basically A and B is there. Then we get A separate, B C complex formation happening. Catalyst transfer the group between the molecules. There will be transfer of groups. You might have heard about transamylase somewhere, or transacetylase. Transacetylase you might have heard somewhere. These two enzymes are simple examples of transferase. Examples of transferase. Then we have hydrolase. What are they doing? You're adding water. Like your amylase is an example here. Amylase is a good example here. Okay. And even you might have heard about pepsin. Pepsin also is a type of hydrolase enzyme. Then you have lyase. Lyase will do what? Lyase will catalyze, break the bonds without using water. Here, bond is broken, bond is broken with the help of adding water. But here, bond is broken without adding water. Without adding water. Okay. Isomerase we have learnt in fructose, in glucose to fructose change. Glycolysis, isomerase. Even in the citrate to isocitrate, isomerization. Then ligase. Ligase is basically joining. Ligase is basically joining. Simple example all of you know is DNA. Ligase is the basic example all of you know. Here is given in your NCERT. Everything is given in your NCERT. Hydrolase, transferase, transferring, hydrolase, lyase. And remember students, whenever there is lyase, there will be formation of double bond. There will be formation of double bond. Okay. Hydrolase, enzyme catalyze the hydrolase of esters, ether or peptide bond. Interconvergence. A ligase catalyze the linking of two carbon compounds. They are joining together. They are joining together. This is your homework question. This is right here is the homework question. Students, I know you might have been like, what about sir? If you have a doubt, how do we contact you? Students, all of you know, you can always contact me on Telegram channel. Just type Basavaraj Sir Biology, you get my Telegram channel. On that Telegram channel, you'll be able to ask me doubts, any type regarding biomolecules. The chapter is done and dusted. There is no more difficulty thing than this. NCERT reading is done. Read the NCERT one more time after the class. Watch this lecture one more time. Any doubt you have, you can always come back to me and get your doubts cleared. Get your doubts cleared. Okay. Now students, all of you know the DNA batch students, and DNA batch I have been doing from Monday. That is on Monday I'll be starting the plant kingdom here. Monday I'll be starting the plant kingdom on DNA batch. In DNA batch we take around 6 classes to finish a chapter. 6 class, 4, 7 class we take. And right on the price is going to be 3999. But if you want a test series, long test series for 1 year plus DNA batch, that is 499. DNA plus year-long test series plus Tatva books. This is the hard copy. This is the hard copy of Tatva books. Then it's going to be 7999. These are different prices of DNA batch, and it is a complete long batch which we are doing. There I have thought, there we show much more visualization. There is quiz, there is doubt solving, and it is going to be on an app. Everything is controlled. Every single day you have 3 classes. First class is around 10 o'clock, second class is at 12 o'clock, third class is around 2 o'clock. 3 classes per day throughout the week except Sunday. Every alternate weeks we have the mock test there. So all that is in DNA batch we are doing there. And students, if you are able to understand today biomolecules, and if you feel that yes, the biggest burden of my life that biomolecules is done from biology, let me know in the comment section. Let me know in the comment section how the entire chapter was. And with that being said, I'll take off right now. Thank you so much, students. Thank you so much. And let me know in the comment section how the class was. Do not forget to like the session and also share the sessions. Okay. And entire notes will be available on Telegram channel. So take care, students. Bye bye and have a great day ahead. Bye bye all of you.